I will go to start. Sir, we will start now.
Yeah, please, yeah.
A very good afternoon to everyone. On behalf of DDH-2020 training team, as well as Director CSIR NIS, Dr. G. N. Sastry, I welcome you all for the Vertical 1 session. The Vertical 1 session today will be delivered by Dr. Prashanth. Dr. Prashanth is a senior scientist in system biology at Birla Institute of Science and Scientific Research in Jaipur, India. He has wide interest on functional genomics, protein informatics, and interactions. He obtained his PhD from Aalborg University in Denmark and gained more than 5 years of postdoctoral experiences. His current research focuses on developing resource-targeted genomic regions of interest via known and unknown interactions, non-coding RNA interactions linked to disease genes and proteins. With this small introduction, I request Dr. Prashanth to deliver the talk on hypothetical proteins as a predecessors of LncRNAs, drug targets on Anvil. Sir, please.
Yeah. Thank you so much for your kind introduction. At the outset, I would like to thank Dr. Narhesh Shastri, who has kindly invited me, and the team hackathon for this. So my talk would largely be focused on hypothetical proteins, their history, and wherewithal these hypothetical proteins could be considered as predecessors of long non-coding RNAs, which myself and our group have been working over the last 4-5 years.
Mute your mic, Nagamani. Just a minute, mute your mic, Nagamani.
It is mute, sir.
There's an echo here.
Yeah, and then in the second part of the talk, probably I'll slightly take over to the affable drug targets that we have recently worked on, COVID-19. So this particular story is really split into two. So if you really see the current COVID times, there is an absolute need for.
Do you have two YouTube videos? Because there is an echo, a lot of echo coming. There is one more device playing in your room, or your.
No, sir. I don't have anything. I'm sorry. I've just cross-checked it. Yeah.
Okay.
Let me see again. I mean, I don't think there is another link. No. No,
okay,
there is another.
Yeah.
So in the current COVID times, probably there is an inherent need to bring affable drugs like what we have seen during the bubonic plague times, where we had a requirement of huge drugs, but there's always a kind of a necessity. What kind of drugs? Are we really looking into the protein-based drug targets, or the DNA-based vaccine theragnostics, or RNA-based vaccine theragnostics? So there are several questions, but from our annotation point of view, the next part of 15 or 20 minutes of the time, I'll probably delve on the hypothetical proteins, which form a part of the known unknown regions in the genomes, and they could probably play a very important role as predecessors of long non-coding RNAs, and these known unknown regions could as well be viable drug targets. So from this picture, as I could see, the targeted one is the drug targets over here, And we also need something called vitamin M, which is vitamin money. So when we say vitamin M, we need an affordable drug in place, during the post-COVID times, because of, huge loss of, economies all over the world. So I come from Birla Institute of Scientific Research. I'm sorry, this is a kind of an self-approbative advertisement kind of thing. I'm a bit obligated to. Ours is not Birla Institute of Technology and Science, but ours is a Birla Institute of Scientific Research, which is based in Jaipur and is a sister concern of Birla Institute of Technology, Ranchi. We also have campuses in Jaipur, and of course ours is a not-for-profit research institute with two departments. One is biotechnology and bioinformatics, and the second one is remote sensing. So our current director is Dr. Punendu Ghosh, and Dr. Krishnamon is head R&D, and we are a group of about 14 scientists and about 7 PhD fellows from two departments, and there are 2 postdocs as well. So we have the biggest auditorium, and we also take the geospatial satellite images to collect the flora and fauna. And the 2 departments, biotechnology and bioinformatics, we collaborate, very often. So my talk would be split into 2 main stories. So the first blue one is about the history of hypothetical proteins. Where are we now? The kind of problems and inherent challenges, and most essentially these hypothetical proteins, how they could probably be playing a role as predecessors of long non-coding RNAs, and most importantly as affable drug targets. I'll try to come up with some kind of case studies, and then I'll quickly go to the curcumin and its role, at which we have recently worked with Professor Kavikishore's lab. And then I'll probably end my talk with some kind of future prospects. So the first part of the talk, since it is called as a training module, so I just thought there could be many students who might really be interested in knowing how these hypothetical proteins are annotated, and what we do, how these known unknown regions could really be identified. But for that, we need to walk through the genomes. So when you have a 6.4 billion base pair of genome, of human genome, not necessarily you need to really walk through the entire intronic and exonic chunks. But if you have a protein of interest, there could be many regulatory elements that are present at the 5' untranslated regions of that particular protein. And then when you walk upstream, you could probably identify whether or not there are any good regulons or regulatory elements that are associated with that particular protein. So the reason why they could be associated with the protein is those are the essential candidates that would probably enthuse you to consider as affable drug targets. So as we know, the holy grailing bioinformatics, until probably 10 years ago, the sequence predicts structure, predicts function, was an absolute affirmative for central dogma of bioinformatics. But commit next generation sequencing since 2009 heydays, we certainly needed to identify not only the mutations, but whether or not any of those particular protein coding genes which harbor these particular mutations could really be considered as affable drug targets. So this is the latest resource. As you could see, we have about more than 160 to 170 million sequences that are deposited in GenBank. And as you could see, the curve doesn't flatten at all. It keeps on increasing. So this is what we call it as an anti-COVID curve. We don't seriously see this particular curve really flattening, even at the parabolic peak. But that's also sorry news, to be very honest with you. It's because we have so many sequences. We need to fight with what we call it as a Moore's law. And we need to ensure that there could be some, meaningful resource, that we could really take up, from these particular sequences. So way back in 1985, as you could see, there were very few sequences, less than about 2,000 sequences, that were deposited. But as you could see, we are somewhere in 2020, so we have really reached a Mammothian peak in terms of sequences and the number of bases that are really being read from time to time, which is quite alarming. So can we really come up with a kind of a meaningful story? Can we say that a group of these particular sequences come up with a kind of a similar function? So that's where the real picture comes into picture. So we have so many of these particular sequences that are available in gene banks. There are three main gene banks, as you pretty well know, the NCBI, the Anvil EBI, and of course the DNA Data Bank of Japan. Earlier, there used to be many express sequence tags, and of course the mRNA floated microRNAs also do exist even today. But the erstwhile express sequence tags were the primary epitome for understanding the known unknown regions, because at that point of time, not many genomes were sequenced. These EST enriched information were really considered to check whether or not any of these particular sequences play a very important role, or any of those particular regions could really be considered as probably small molecular targets. So given this functional and evolutionary information that is always really challenged, not all sequences converge and produce a good number of structures. So if there are a loss of structures, then there is always a loss of function. And unfortunately, if there is a loss of function, then the so-called affable drug targets is also juxtaposed in question. So we don't know whether or not we are really identifying affable drug targets. So having said this, so there are many known unknown regions. When I say known unknowns, I have a slide in the later part of the presentation. The known knowns, as per the Logan's concept, David Logan, he conceptualized the theory of the known knowns and the known unknowns. The known knowns are nothing but the first known could be a protein or a gene, and the second known could be a function. So if we say a protein that is known and the function that is known, which means the protein is well annotated and probably well curated as well, and that is where you have in the real sense you have something called the ref-seq sequence or a reference sequence, okay, which is a highly curated one. Whereas the known unknown regions, you know that by virtue of your annotation that a protein exists, or maybe the expression of that particular protein by virtue of your annotation is known, but function of it is not really known. So whether or not it is really expressed is not really known in situ or in vitro or in vivo. So these are the known unknown regions, and these are nothing but the hypothetical proteins. So usually when the genome is sequenced, you get a lot of these hypothetical proteins. Many of these hypothetical proteins in humans, they start with something called XP underscore, which means they are simply predicted, and after second or third time annotation, they're simply tagged as NP underscore 00, which means this particular protein is just annotated, so which is just a known protein, and that particular protein function is not yet deciphered. So that begs a kind of a question whether or not any structural repositories do exist for hypothetical proteins. So yes, there are a couple of repositories, but this particular work was conceptualized way back in 2004 when I started my PhD and when I finished it in 2008. So there was an inherent need for development of a hypothetical protein database, and that was how we started. So we needed to carefully check what are those particular non-repetitive sequences or the non-redundant sequences, because there are many redundant sequences and many chunks which are misannotated or which are really poorly annotated. We need to really take that particular entity and transform into a real biochemical knowledge. So the challenge here is not just the sequence predicts structure, predicts function, but also the real sequences itself. How many of these sequences are really bona fide? How many of these particular sequences would really start with the methionine as a start amino acid? How many of these particular sequences would start probably with leucine as a start amino acid, which many of these particular bacteria take up as a start amino acid sites? So there were a lot of questions, and there was no the real picture that were really considered. So the real challenge at that point of time, or even today, if you really see, excepting that black one, we needed to ask, where are these hypothetical proteins localized? What are these particular domains that these hypothetical proteins harbor? There are some important domains like the Kia domains. There are also some important domains like the DUFs, the domains of unknown function. So these particular domains are absolutely known in eukaryotes. Whereas in bacteria, there are also some Kia domains which are not really recognized. And these particular hypothetical protein candidates do exist across the length and breadth of different bacterial species. So we then check for the native structural analysis. Of course, we simply predict what are the structures that these particular sequences make up. And then we check the nature of that particular protein. We might go for LCMS or GCMS in a rare case, to characterize them, and then check their pathways and then map up with the ontologies. But the real question that really comes into picture today is, what are these particular proteins interacting with? Are there any known proteins to which these hypothetical proteins are interacting with? So if a hypothetical protein is known to be interacting with a known protein, can we transfer the annotation of that known protein to this hypothetical protein? So that was what we asked. And that is what we call it as modeling this particular protein, using a training module. But interim, we only knew the known knowns, the known unknowns. But during this particular process, there is also another third category, which comes by virtue of, say, a kind of a pull-down assay that you probably want to make. In an experiment, say like a co-immunoprecipitation or a Western blot, or when you even run a kind of a MAGIT or CELDIT of the huge proteomics-based analysis, there could be the unknown knowns. So which means you don't know what is that particular protein, but all of a sudden you have some functions deciphered. Okay, so these particular things also really pop up, and all these things we could probably take up as a part of our training module. But to develop this particular model, how do we conquer it? So we have these so-called hypothetical open reading frames, which are also absolutely seen in SARS-CoV-2 as well. Just like SARS-CoV-1, the erstwhile 2004 SARS-CoV-1. And now we ask which of these particular 6 reading frames is really translated. So we have 6 reading frames. Many a time a wrong reading frame might really be translated, and conquering with the so-called hypothetical protein. But thanks to stringent algorithms that we have today, like T-BLASTX or the MEGABLAST, sometimes BLASTX as well, which really allows us to even go for the bidirectional best BLASTX and check whether or not whether or not the hypothetical protein is really translated from a real reading frame, not an unreal or hypothetical reading frame. Of course, we check that codon usage. And then the most important thing, this is what we call it as genome walking. So in genome walking, where the protein exists, that particular gene. You walk upstream of that particular 5' UTR or 5' untranslated regions. And the very first thing you really find is, so from your window, if this were a protein, the very first entity that you probably really find is the transcription factor binding sites, because this is the candidate which has got the highest influence on this particular protein coding chain, how this is really transcribed. And then further upstream, you have got the long non-coding RNAs. You'll have promoters in between them. And of course, at the end, you have the poly A tails. You've got some regulatory elements like the open chromatin elements or the CTCF motifs. You've got some enhancers. Many a time, these enhancers are really bound within the open chromatin elements all through the length and breadth of that particular protein coding chains. And most importantly, even if your particular protein is, say, it has got say 19 exons, so the protein coding chain has got 19 exons, you have got the long intergenic non-coding RNAs, which means they are sitting within the exonic boundaries as well, within the intron exonic boundaries as well. That is the reason why, as you could see, we put it as in red color. So you also find the LncRNAs. So this is how we simply walk through the genomes. We check for the signature sequences. We walk through the genomes. There could be any browsers. Here I've shown you the UCSC Genome Browser, which we walked some 8 or 9 years ago. But thanks to the wonderful browsers today, we have got the Ensemble browsers as well. And then we check what and how these particular random sequences that are bound within these exonic boundaries, whether or not they're really making any true sense of those particular sequences, whether or not those particular regions are really impromptu present or not. So we check what are those short intergenic and the long intergenic boundaries. And then we ask, now that we have found one hypothetical protein and we have some in the 5' UTR, you have got a lot of regulons like the enhancers, the open chromatin elements, the transcription factor binding sites, the promoters, the long non-coding RNAs, some other non-coding RNAs like microRNAs, in a rare case, the competitive endogenous RNAs, the pvRNAs. So all these kind of so-called regulatory elements have the highest influence on the way this particular protein is associated with, or the protein coding gene is associated with. So these are the real regulatory elements which we could really consider them as characteristic biomarkers. So they are the candidates, they are the real guys, which we can really say that they can be considered as the elements for the drug discovery. Okay, so and now comes a very important question: where do we see these applications? So in diseased conditions, so if you really take the human diseased phenotypes, say in cancer phenotypes, what would be the fate of these particular regulatory elements given this particular disease gene? And what would be the case in a normal or in a healthy being? The same is the case with plants in case of trait biology, where you can decipher the environmental association. So at the end of the day, what really matters is saying ohm to the function. Ohm is nothing but many, okay, or monies. So you always need to talk about the genome. So you've got many genes, many regulatory elements, many interventions, because this protein coding gene is always associated, is always dependent on how good this particular gene is really associated with these regulatory elements. So, and these regulatory elements could really be better used for training efficiency, for making good number of classification algorithms, or making some kind of machine learning heuristics. And then we can give an approximate functional distribution. Then we can say that yes, so many proteins could really be localized to so and so organelles, and they might be associated with different pathways. So so this particular information can of course be extrapolated with different algorithms. There are different predictors, different subcellular localization predictors. But first one is the hypothetical protein sequence, and then comes whether or not that particular protein sequence has a 3D structure associated with that. If not, can we infer or can we model any structure for those particular hypothetical proteins? Can we infer any kind of hypothetical protein structure, that could be associated with the known structures that are deposited in say protein data bank? If not, how do we take along? we know as structural biologists, many of you must be knowing that we have a huge crunch of structures which tags down to a ratio of 150:1. So which means for every 150 sequences, probably you have one particular structure that is deposited in GenBank. Then how do we really take along? What if if you are a simplest functional genomicist like me? What if you're not a structural biologist? So this is where the intralog mapping comes into picture. So intralogs are nothing but in one organism you take one particular protein that is known to be interacting with, say, protein B. So the homolog of this particular protein A would be, say, A dash in other organism, and the homolog of B could be B dash in other organism. So these are nothing but the orthologs. So since these proteins are really interacting with each other, we might also tend to say that the orthologous interacting partners could also be interacting. So these are nothing but the intralogs. That intralog information is also taken into picture, and then we can make it. But there are some fallacies with this, because you have a lot of time associated with this, because you need to probably refine the structures. You need to check where exactly that particular protein is really bound to. In case of checking that particular site as an affable drug target, you need to probably see whether or not that particular bound region is really associated with any folds, or whether or not it is really exposed, because buried surfaces may not really play a very important role. So that's where the transitive property might also play a very important role. So in a transitive property, just like intralogs, in intralogs you talk about orthologous interacting partners of the protein interactors of that particular organism. Whereas in a transitive property, if A is interacting with B and B is interacting with C, we assume that A could also be interacting with C. Now comes a very important question. When we say interaction, as a systems biologist or as a genomicist, I would probably talk about interaction only in terms of physical interaction, which means impromptu, in situ, in vitro, these must be physically interacting. So these are not associations, by the way. So when we say associations, they might have a similar domains. They might do some kind of similar functions, but not every time. But you always need to talk whether or not those particular proteins are physically interacting. Okay, and that would really give out this sequence to drug target transformation. So that begs us a question. Now that we have some hypothetical proteins. We know how to annotate them. What are the candidates that could be enriching for a hypothetical protein coding gene? How do we walk and all those kind of things? Could these particular hypothetical protein annotation from systems point of view could they really make any kind of studies for drugomics? So can we really consider any of these particular defined targets using systems biology approach for inferring drug targets? So where are these applied? I think we have got wonderful proteogenomic approaches. We have got various multi-omic and systems and synthetic approaches, which we have dealt in our previous reviews as well. This is where we have exemplarily shown some of the candidates, how some of the hypothetical proteins could really be used as candidate interventions for drug targets, if not directly for drug targets, but candidate interventions. So from 2006, I believe, 2006 to 2020, we have about 10-11 publications on hypothetical proteins, which produced about 4 PhD fellows, including me. I myself and my PhD fellows also have done some annotation as well. But it was a very enticing experience. It was an absolutely wonderful experience. But then we asked, are we really drinking an old wine every time? I mean, drinking the same story, bucking up the story, because hypothetical proteins are, of course, they keep coming every time. Even if you sequence a genome, you'll have these hypothetical proteins every time. And then came a story which we deciphered. We developed this database thanks to Sundar, who is my mentor as well, who works for a government based data science ministry in Singapore. He was in South Africa then in 2009, and together we developed this hypothetical protein database, which is hosted in BioClue's website. And we found, as a part of my PhD, we found hypothetical proteins similar to MLH1, which is a mismatch repair protein, which I'll be talking in the next few slides. But one of these particular applications of these hypothetical proteins, which Girik also has wonderfully attempted here, is there's also something called matchmaker. So if you simply give a hypothetical protein, it will tell you whether or not that hypothetical protein could really be considered as a potential biomarker. And it will go through certain vector-based decision trees. And then it will tell you whether or not those particular hypothetical proteins could really be indeed as considered as potential biomarkers. So when we say biomarkers, it's only biomarkers. These are not really affable drug targets per se at this point of time, because any drug-based, delivery-based approaches need rigorous structural docking-based approaches, which unfortunately I'm really sorry that I'm sorry if I'm disappointing you. I'm not a structural biologist, but we have wonderful leads in this particular case, where we can say that these hypothetical proteins could really be considered as affable drug candidates. So this is how we started. We thought that we're always drawing an old story of hypothetical proteins. But over the last 3 years, Girik Malik, who is currently in Harvard, USA, and Tanu Agarwal, who is in Jaipur-based clinical research company, they have wonderfully worked on this particular exciting story. Earlier, we have developed something called Hypotome. Hypotome is an interactome of hypothetical proteins. These are the real candidates where we can say that if there is a hypothetical protein in the genomic loci, would that particular hypothetical protein tend to be expressed across different tissues? Because this can be checked from various RNA-seq based studies that people keep doing it. There might be some co-immunoprecipitation. There might even be virtual pull-down assays that people keep doing it. We keep a watch on this, because in humans there are about 1,100 hypothetical proteins as on today. So these hypothetical proteins, 1,100 hypothetical proteins, who knows? They can really play a very important role. They can really come up with a candidate, drug pathways, and this is how we walk through and check from our annotation. So this is what we do. When I started my PhD way back in 2004, there was no rigorous mechanism, although I was a Perl and Python coder. I would probably code for first 100 or 200 lines, and maybe from 201st line probably I would faint. So I was not really a rigorous coder. But what we did was we checked each and every hypothetical protein, taking the consensus from these 3 gene banks, went through this UCSC genome BLAT and NCBA BLAST, checked for the percent identity, annotated them, each of them, checked for the bidirectional best blast hits, and developed a hypothetical protein database. And this is how we developed a kind of a hypothetical protein interactome entity as well. And this was a huge hit. And then what we did was, since I was also working in the wet lab, we found a candidate protein called MLH1. MLH1 is actually a DNA mismatch repair protein. It is localized to nucleus, but in mitochondria, if MLH1 fails to repair, because there is no mitochondrial MLH1 per se. So if DNA mismatch repair fails inside the mitochondria, then there is an SOS, okay, and then the mitochondrial proteins makes a huge shout, and then a protein from nucleus here, the MLH1, which is a mismatch repair protein which is localized inside the nucleus, comes to the rescue of mitochondria, does the repair and goes back. So this is what we call it as a piggybacking mechanism, and this is what we learned from my mentor, Lina Rasmussen's lab, where I did my PhD. And we checked across different interactomes, checked various candidates, and then we found that there's one hypothetical protein, which we call it as EAW74251, which still exists as a hypothetical protein, which probably does the function of this DNA mismatch repair protein. And this was the first interactome which we developed in 2004, even when Cytoscape was not there at that point of time. And then, as you could see, the yellow ones are all the mitochondrial proteins, the red ones are all the disease proteins, the bold ones, the circle ones are all the proteins of our interest. And then we asked the algorithm, come on, you have the protein candidates within your vicinity in a virtual cell. Now you keep interacting, how these particular proteins keep interacting. And then, as you could see here, we get a lot of interacting partners. So some of our proteins are interacting with the mitochondrial ones. And as you could see here, this mitochondrial queried one is interacting with a deceased protein here, as you could see. In fact, this deceased protein is nothing but a protein that is accurately used as an affable drug target. So this is where this particular protein is. But in some cases, we also have protein interactions between a deceased protein and a mitochondrial protein, which is also a deceased protein. So we have several euphemerisms here. And then we checked, these are only the first grade interacting partners, which is the first interacting partners of your query. And then when we expanded it, you see, the real picture, it keeps on interacting. And as I said, there was no visualizer. I really love this slide. I really keep telling my colleagues, that this was certainly not an easy way to do. But, as you could see, this is how, we have written it with a pencil and checked what is that particular protein. And we did this. And the protein network really expanded. And as a result, we found another hypothetical protein from virtue of our interactions, which is a DNA repair PARP protein, which is a poly-ADP ribose polymerase protein. And incidentally, this particular protein over the last 10 years has really been used as a drug candidate for treating DNA repair mechanisms, not only in mitochondria, but also for other diseases like skin rashes and other things. And the drug name is rucaparib. So you see, this is how the translational value of these particular proteins as affable drug targets are really taking place. And as a part of my first postdoc in 2010, I was really piggybacking between Hyderabad, Boston, and Denmark. My first postdoc in 2009 and 2011, we worked on something called Aptamers. So Aptamers, many of you as chemists, you must be pretty well knowing that Aptamers are small molecules. They could really play a very important role for specifically targeting to a receptor, and probably they could really block the real or the natural ligand binding mechanisms, and probably they can really play a very important role. And then we asked if any of these hypothetical proteins could really play a very important role as aptamers. That story, which is still evolving, I'm working because in developing or in synthesizing aptamers, we need selects-based strategies, and this is what we are currently working. And thanks to Professor Giri and Professor Kavikishore sir, with whom we are collaborated on this, and my wife Renuka is also synthesizing these aptamers as well. So I'll not bore you much on this, but the most exciting story here that I wanted to delve here is, can we really say that if there is a hypothetical protein as a candidate, at any point of time, if it were to be misannotated, can this hypothetical protein could really be intron to be coming from a intronic chunk, and then that could be considered as a say as a non-coding partner, or maybe by virtue of online or automatic annotation system, you simply characterize a protein as a hypothetical protein. But after curation, you realize that that hypothetical protein is no longer hypothetical, but it is really coming from the non-coding chunks. So this is how the story began. So as I told you, Girik and Tanu, they have excellently worked on this. And then, as you could see, the pink ones as over here, the purple ones, I apologize, the purple ones here, from the hypothome which we have developed over here, these are all the candidates which could be considered as affable drug targets. And this is what we have developed, and this was published in Current Genomics recently in January 2020. So basically, there are many hypothetical proteins that are annotated. But there are some misannotations, but many of these hypothetical proteins are really indeed the predecessors of long non-coding RNAs. So even before a long non-coding RNA is really known in the organism, you may say that it can really be a good candidate. And this is another exciting study which I was working for Novartis, and I've taken the prior permission on this before I inserted this particular slide. So they had a lot of exciting stories. They wanted to take from budding yeast, from Saccharomyces cerevisiae. They wanted to know how many of those particular proteins are indeed really be interacting with the hypothetical protein. So we applied our hypothome approach. We identified a candidate, which is HSPC148 over here, and then we did our annotation. So we used a lot of annotation-based strategies for this lysosomal protein prediction. We came up with a good strategy, but our strategy is again simply based on that annotation schemes which we have developed over the last 13, 14 years towards that. So the first part of my particular talk, so there could be the known unknown regions, they could be non-coding RNAs. So many a time these non-coding RNAs could also have coding potential, and many of them, they play a very important role as regulons or regulatory molecules. They can be playing a very important role as effective biomarkers. Many a time this coding potential of non-coding RNAs could also be, in principle, be playing a very important role towards interacting studies, which I was telling you about the transition properties and also the intralogs. And of course, our hypothetical proteins are predecessors of long non-coding RNAs, and how they can really play a very important role as drug candidates I have discussed. So the part 2 of the story, this is a recent thing, sitting at home. We were obligated to sit at home with very weak servers, with not so good efficient servers at home. But of course, we had our command line access to all these kind of things, which we did. And we did a kind of an exciting story because SARS-CoV-2 genome was out at that point of time, and then we asked the very same question. We walked through the 29.9 kb region of SARS-CoV-2 and asked how many of these particular regions, have a very important role, towards determining, antigenicity. Okay, or the other way around. Can we say that these candidates, when we walk through the genomes, how many of these particular regions are really opening up? How many of these particular regions are at exposed surfaces, which would really help us, move forward in identifying, probably, epitopes, or probably those particular regions that could really be targeted, for aptamers or antibodies. Okay, so this is what we did. So I don't want to again bore you with SARS-CoV-1 and SARS-CoV-2 story, But my point at this point of time is we have many structural proteins that are associated with SARS-CoV-2, which we call it as MENS, the membrane envelope, the nucleocapsid and the spike. But to us, there's also a very important protein called the non-structural protein, the NSP10, which plays a very important role because we have recently shown that this is the candidate when enters the host, the ACE2 takes the control of this particular virus, or the other way around. The virus takes the control of this ACE2 because ACE2 is the receptor, along with TMP RSS2. Apart from that, there are many hypothetical proteins sitting within the vicinity of this particular mitochondria. There are many interleukins. Okay, so these candidates, what they do is they take the control of this particular virus, and then a big transformation takes place. So how many of these particular candidates, like the non-structural proteins or the structural proteins, would have an influence of that particular receptor? So that is a question, which probably I'll just talk to you at a later point of time. But this is what, we have done work with Professor Kavikishore sir and Professor Giri, and as well as Professor Govindarajan Padmanabhan of IISc. So we did a kind of a comparative docking studies on curcumin with COVID-19 proteins. It's still under review, unfortunately. We have taken the community annotation, we have taken the critical comments of various people, and together we are publishing this. So we did a simplest docking. We have not only delved into those 4 structural proteins, but we've also taken the non-structural proteins, the NSP10, because NSP10 is also considered as an affable drug target. And interestingly, we compared this curcumin with all the well-known 13 commercial drugs that are available in the market, including the hydroxychloroquine, the remdesivir, and it fares absolutely very well, albeit the fact that the bioavailability of curcumin is very low when compared to the other well-known commercial drug targets. And then we also asked whether or not this efficiency in terms of less concentration could really lead to consider curcumin as a potential antiviral against SARS-CoV-2. So that is what we have done, and currently we are coming up with some kind of simulations, which our collaborators are really working on that. So being a not-for-profit institute, we don't have any huge GPU-based servers, for simulations. That's unfortunate, but we are just not trying to take help of our collaborators in coming up with that particular story. But someone like me as a HP researcher, hypothetical protein researcher, again, our target, our focus again on hypothetical proteins. We asked a very simplest and pertinent, is only ACE2 as playing a very important, and other particular proteins which might be waiting in queue, maybe in the cell, ACE2 might only be receiving the virus, but at the back of it, there might be some other proteins which might be waiting in queue. क्या होगा अगर ACE2 अगर नहीं लेगा, अगर ACE2 अगर इस को नहीं लेगा, what will happen to that particular proteins that might be pending? Of course, we have the transmembrane proteins and all, but we also have an interleukin 17A, which was a kind of hypothetical protein until a couple of months ago. That could really play a very important role, and it can be considered as a master regulator. And recently, it's a patent that has really come up into picture. This is an interesting and developing story over the last 2 months. AstraZeneca has come up with this patent with Ixekizumab, which is a humanized IgG4 monoclonal antibody that they have developed against this interleukin 17A. But they also targeted these hypothetical proteins. Okay, and it has absolutely been known be implicated in variety of diseases, including arthritis, the plaque psoriasis. Even there are some symptoms, with SARS-CoV-2 patients, with psoriasis and lack of, taste and composure and all those kind of things. So this particular candidate drug has been used as an affable drug target. So the final part of my particular talk. In fact, when we talk about hypothetical proteins and all, a very important question comes into picture. You might ask me a very pertinent question. Prash, we're talking about hypothetical proteins, maybe linked to non-coding RNAs. Can you come up with saying could really be used as targets? Because the life of any RNA in a cell is very small or is very less because they're absolutely labile. We say that these can be important, but our annotation-based system, we have developed a kind of a 9-point classification scoring scheme, which Johnny Ijak, my PhD fellow, he has developed over years. So what we have done is we have checked the exact site zones of these hypothetical proteins that are absolutely be associated with long non-coding RNAs. So the long non-coding RNAs are absolutely evolving. Many of those particular candidates are developing. So the site at which the long non-coding RNA is probably associated with the hypothetical protein could really be very specific. Those are the candidates which play a very important role for drug targets. And of course, the story too. This is what we are coming up with a story. So these are the aptamers coupled with the lateral flow assay. A simplest lateral flow assay. So you have one aptamer that is inserted over here. The aptamer would really come at the end of this particular streptavidin-based bead, and then where you have got this FITC. And then at this particular lateral flow assay, you just insert your sample, okay? And once you have that particular sample, mixed with this analyte, then you will be able to know whether or not, with a change in the color, you'll be able to see whether or not a particular person is COVID positive or COVID negative. So this is also Professor Giri sir is really coming up with it, but I designed aptamers length and breadth, and probably it's a developing story at this point of time. And of course, the third story, which I didn't describe because it doesn't make any sense for this hackathon-based done recently was with Professor Keshav Singh. We have identified certain mechanisms on how SARS-CoV-2 could really deter the acids in the body. So what happens is SARS-CoV-2 it attacks mitochondria. So for a healthy, there are about 2,000 mitochondria per cell. But this SARS-CoV-2 high mitochondria, that high chain is associated with mitochondrial DNA, but also these candidate long non-coding RNAs as well. And these long-coding RNAs, interestingly, these are highly expressed in prostate. Is also coming up with a kind of a story. So these long non-coding RNAs is 4 long non-coding RNAs, which are the non-homozygous brain pan, Roche, and Novartis. The final part of the conclusion, we need to bring transformation of understanding the antigenic determinants We need to understand what are those exposed regions, whether or not those exposed regions could really play a very important role in considering them as good epitopes. Of course, we need to develop annotation and curation-based strategies for development of kits. And for considering these hypothetical proteins as affable drug targets, we need to know the stringent known unknown regions. We need those raw sequences. In case of SARS-CoV-2, labs like us, we're not that rich, but we don't have raw sequences, for all the 981 sequences that are deposited today. So we need raw sequences because as a systems biologist, we need to assemble these sequences ourselves and then understand whether or not they're really assembled properly or not. And then these sequences keep changing, they're variable. And of course, these hypothetical proteins, they are not only used as predecessors of long non-coding RNAs, they could probably be used as aptamers and probably Aptamers or nanomers as well, which could really play a very, very towards my end, I think we have come up with a Galaxy India project, where Anshu is really taking this, Anshu and Harpreet. But we have a big person behind this, Dr. G.N. Shastri. So Sir is with us. He has been a guiding force for this. From Northeast, we also have Devendra Biswal, and we also have Sangram, who is really taking this. So please join this Galaxy India initiative. We at BioClis are also, a part of this. So the works which I've the two stories. The first story about the hypothetical proteins as predecessors of long non-coding RNAs was by Greg Malik, who is currently in US, and Tanu Agarwal. Second story, of course, the hypothetical protein annotation and all was done by my erstwhile PhD fellow Johnny Ajak, who is currently a postdoc. My other PhD fellows and postdocs, so-called visiting postdocs, of course, Nidhi, who is a PhD fellow, and Ayam, who is also a PhD fellow, Harshita, Purva, Anchita, and Sonal, of course. So these are the people behind this. And of course, Obel was also a part of this hypothetical protein aspects of long non-coding RNAs. The medical genomics team, of course, Keshav, Gyan, Umakanga, and of course, my mentor KM and Praveen Mathur, who are a part of this team. The Karthaman story is largely done by Abhinav and Renuka. They're excellent structural biologists. They have been working on this. And of course, this would not have happened with these four people, Professor Kavikishore, Professor Giri, Professor G. Padmanabhan, and Dr. Nagaraj. And the machine learning takes trying to work with Pritesh, Jayaraman, and Sundar. So thank you so much. I'll be happy to thank you so much. And team Shani and Uday. Yeah.
Hello, sir, am I audible? Hello.
Oh, not alone. Hello.
Hello, sir, am I audible?
Hello. Yes.
Okay.
Yeah, you are audible, please.
Thank you, sir, for your informative talk. Sir, we received few questions. Due to time constraint, we will take 3 to 4 questions, sir. So may I, sir?
Yeah, please. Yeah, please. Yeah.
First question is, in your study, which algorithm was used to study the protein-protein interaction network analysis?
Yeah, I mean, we have a 9-point classification scoring schema, which we have earlier called it as a 6-point classification scoring schema. So what we usually do is we always take consensus. We take, gene mania, string, and then reach consensus. And again, when we visualize it, we also do visualize with a Visant, which is a kind of a visualizer, and also Cytoscape. and also osprey as well. So for example, that particular interactome that which we have shown with hypothetical proteins and long non-coding RNAs, there is a visualization from osprey. So we always take consensus. It's a mixture of, it's a conglomerate of gene mania and string. Yeah.
Thank you, sir. So next question is it possible to trace the changes in RNA through structural biology?
Yes, that's indeed a very good question. We can. Another part of the story which Sonal and Renuka are working on it, where our work is under review. So what we tried to do was we checked certain regions in the RNA. We checked how many of these particular RNA bases would really be impromptu be docked, because those are the particular regions that are present as the exposed surfaces. We can openly say that they could be the candidate bases, but it needs certain kind of an experimentation in the lab before we consider them as viable candidates. Yeah.
Thank you, sir. So third question we have, what is the function of regulatory elements?
Yeah, regulatory elements, as the name sounds, they regulate the gene expression. Okay, so what they really do is, these are the candidate elements that are usually present upstream of a gene. And the very first regulatory element that we can really think of is the transcription factor binding sites, which has an influence on how that particular gene is really transcribed, how that particular gene is transcribed to mRNA, and probably later, processed to protein. So the first one is the transcription factor binding site, maybe the promoter, that will be followed by the enhancers. And these elements together, we call them as the regulons. But the most important element could also be the open chromatin elements. They play a very important role. So in a nutshell, regulatory elements are those particular candidate regions, preferably in the untranslated regions of the genome, which play a critical role in the way the gene that is present in the downstream of that particular region acts on. So in order to ensure that the gene is functionally written well, these regulatory elements play a very important role. Yeah.
Thank you, sir. The fourth question we have, what kind of structural changes you have observed in NSP10 after docking?
That's a good question, but I really don't remember. But it's not about the structural changes. But what we did was when we have taken NSP10, we also have taken NSP7 as well. NSP7 didn't gel well with curcumin, but NSP10, outstandingly played a very important role. It could also come in close proximity with curcumin. Although there was a big time lag when compared to the other structural proteins like the nucleocapsid and the spike. But 10 was also there, always a bit ephemeral. It's also it's a complete interaction. It's only kind of a transient docking. Yeah, you can kindly go through that particular paper, please. Thank you.
Thank you, sir. So last question is, sir, can we design aptamers that can wrap to spike proteins and inactivate the viral infection? What is your viewpoint on it?
Yeah, I think that's indeed a good question. In fact, I think there are many papers that have really just started coming up over the last 2 weeks. One of the important drug candidates that probably Bharat Biotech is also coming up with is also this aptamer-based strategy as well. So in fact, aptamers have been absolutely viable players because they are cheap. I mean, I don't say that they're probably cheap these days because they have also come up with par ways. But at least they're specific, they're long-lived, they are actually binding to those particular regions which inherently can be pulled down from the entire regions of the structures, and most essentially, if we are designing an aptamer for one particular intended region, like in this case a spike protein, not essentially this particular aptamer that is designed for spike could really be used for that SARS-CoV-2 as well. So maybe in the near future, if at all, if there is SARS-CoV-3, that might really come into SARS-CoV-1 or MERS or whatever other entities, the aptamer that we have designed could slightly be changed, because we know that spike is the inherent protein that is really regulating the infection, and that could be primarily be used. But it all depends on how viably these particular aptamers are really used. So, for example, in the case of lateral flow assay that we have used. But it's a kind of a developing story. But I see that aptamers could definitely be used for the spike proteins, but my best bet would be aptamers for NSPIC10 as well. So this is what I thought of.
Thank you, sir. Thank you for answering the questions. On behalf of DDH training team and Director CSR NACE, Dr. G. Narahari Sastry, we once again thank you. We would like to thank you for accepting our invitation and delivering your lecture in DDH training program. So I hope your lecture would definitely help the students and motivate and encourage the students to participate in Drug Discovery Hackathon. Thank you once again, sir, for being with us. Here we will wind up the Vertical 1 session, and there is one announcement for the participant and viewers. Please attend the Vertical 2 session from 4 p.m. Thank you. Thank you, sir.
Yeah, thank you. Thank you so much. Hima, Abhishek. Thank you so much.
Thank you, sir.